Okay, is this thing on? Haha, all right. So I'm really feeling it. I'm on a roll today. Okay, so Grace is asking for RNA processing, RNA splicing, and gene regulation, upregulation, and stuff like that. Okay, so this should come to remind you about lac operon and trip operon. Okay, so Thanksgiving time, we got the milk and we got the turkey. Okay, so the difference is that the lac operon is permanently off, and if you drink milk, we got to turn it on so we can break down lactose. Okay, so it's permanently off, but the presence of lactose turns it on. Now the trip operon is always on, making tryptophan, an essential amino acid, but it is turned off when you eat tryptophan. Now, there's not a lot of tryptophan sources in terms of proteins, right? It's like all exciting that it comes in turkey. It's not something you would get necessarily every day, but it is something, an amino acid that's necessary for proteins. Therefore, you need to have that always running, okay? Now, what does that mean running? What are we making? We're making a protein, okay? How are proteins made? Proteins are made by the ribosome translating the mRNA, which was transcribed from the DNA, okay? But in order to transcribe an mRNA from the DNA, the RNA polymerase has to land on the operator region of the DNA, okay? So once it lands on, and I'm scrolling through some pictures here, which would definitely help, and once I get a little more technologically savvy, hey, take it easy, okay? Well, I grew up when it was dial-up, all right? You don't even understand. So I'm not linking no pictures or videos or anything like that. All you got to do is use the Googles, okay? Look up lac operon, look up trip operon, look up RNA processing, and then look up alternative splicing, and this will serve you very well in this little lecture. Okay, so the deal is that, yeah, we've got the promoter region of the DNA, and part of the promoter region of the DNA is the whole thing is called the operon. Within the promoter, there's a region where the RNA polymerase would like to bind, but if something's blocking its way, he keeps bouncing off and he can't bind. Okay, so this would be the case of the lac operon, which is permanently off. Okay, so RNA polymerase can't bind, and therefore it's not going to run transcription because there is the blocker. Okay, which is literally a protein that is latched onto the promoter region of the DNA. Now, this protein that's latched on is called the upper repressor. Okay, it could be a repressor, and it will be locked on to the operator region of the DNA within the promoter, okay? Now, how do you get this guy off? Well, all you got to do is have lactose there, okay? When you digest lactose, and then it's converted into allolactose, so just like a simple little conversion, then that thing goes and binds to this repressor protein and click, falls off. Then RNA polymerase, who's been trying the whole time, gets to jump on that DNA right at the operator region and chug transcription in order to make that mRNA, okay? And what does that mRNA end up making? It ends up making a combination of proteins that will break down lactose, okay, which is a sugar that is not always present in the diet. So you don't want to be making those enzymes lactase in order to break down lactose, unless of course lactose is present when you're like a little wee mammalian babe and you got milk in your life, okay? So then once you pass the infancy stage, that repressor is going to clamp onto the operator region and prevent you from breaking down any type of lactose, and it's more of a permanent fix there. That's called lactose intolerance. Okay, so the other one is the trip operon, which is permanently on, making tryptophan, an essential amino acid, in order to make proteins. But in the case when you eat it, you don't need to make it. That would be a little redundant, right? So the presence of tryptophan should put a blocker on the operator region of the DNA, okay? Preventing RNA polymerase from going. So RNA polymerase is usually making the mRNA, which makes the proteins that make the tryptophan. Bing, bang, boom, go, central dogma of biology, right? But then when tryptophan be coming in, is going to latch onto the repressor, activate it, and say, clampity clamp. No more time. No more locking on for RNA polymerase. No more transcription. No, no, no. Okay? And so that's called it is a inducible operon. A repressible operon, because you can turn it off, even though it's usually on, tryptophan. Whereas the lac operon is called inducible operon, because you can turn it on with the presence of whatever you need to break down. Okay? Now, connected with that is the concept of RNA processing, which is pretty simple. What you got to do, or what you got to know, is that the DNA to mRNA, which is eventually then translated by the ribosome into making a protein, there's a lot of junk DNA in our original thing that we're going to transcribe. Okay, so RNA polymerase just goes through and it's like, A's to T's and U's on A's and, C's on G's and G's on C's. And it's going to make that mRNA. Now, some of that stuff is just excess junk that evolution has just kind of accumulated over time, right? So it's like, there's a little bacteria piece here. Oh, well, we used to make it like this, so here's the instructions for that component of the protein and whatever. So like, there's some extra stuff that like, we gotta splice it. We gotta cut out certain spots, and then we gotta splice the rest of it together, and then we have our finished RNA piece. This process is called RNA processing. It's like the simplest name ever. It's real nice. So the first step, of course, would be transcription. Go from DNA to RNA. They call that pre-mRNA, right? Then they're going to put a cap on it. Now, the cap goes on a 5' end, okay? And that's going to be like the head, the top, right? And that's where the ribosome is going to start, because the ribosome is the only guy that goes backwards from 5' to 3', if you remember that. After you put the 5' cap on, you got to excise, or you got to pull the introns out, and then you got to splice the exons together. So I like to say the ins go out and the exons exist. They remain. Once you splice those exons together, you have your 5' cap. Then you got to put on your poly A tail. Polyadenylation means you're going to be putting many adenines. Should be putting extra adenines on the tail end of things. Now, why protection? Okay, to make sure when that thing goes outside of the nucleus to go find a ribosome, he doesn't get busted up on the way there. So we got a little hat and a tail just to make sure that he makes it there. That's called RNA processing. Now, the cool thing that Grace asked about today that I got to share with you is something called alternative splicing, which basically means that a section of DNA, the splicing I just told you about, the RNA processing I just told you about, that can be done in a number of different ways to create different proteins. So one particular section could have, this exon, this exon, this exon, this exon, this exon, this exon, and a whole bunch of introns in there that are the junk. Now, maybe if you take out all the introns and then you put all the exons together, you make the full version of the protein, okay, call it protein A. But maybe if you take all the introns out and you leave most of the exons, just this one, and then this one and this one, but take those exons, let's take those out. If you put those together, you make a smaller, different functioning version of the protein. We could call it protein B, right? Or A1 or something like that. Now, it's going to be a different shape. It's going to be a different size, and so it has a different function. It might be related. It might work well with protein A. Who knows, okay? And that could be done a multitude of ways. So this one section of DNA is not just responsible for making one type of protein. This alternative splicing says that you can read different instructions to make different things. Now for the analogy time, right? So it's like reading a recipe on how to make like a triple layer, German chocolate, cheesecake layered, with fresh raspberry filling kind of a thing, right? Now, You could follow that whole recipe if you want to make the Mac Daddy of desserts, right? That has raspberry filling and a cream cheese frosting and a cheesecake layer and a German chocolate layer and, you know, all this stuff. Or maybe if you just want to make the German chocolate cake, you don't have to read the other instructions on how to do the raspberry filling part and the cheesecake part. And maybe you read just the part. So you're like picking and choosing which part of the recipe you're reading and putting together. Right? Whereas like and then you could also just make like you could do, all right, well, I want to do the raspberry filling and the German chocolate, but I'm not really a fan of the cheesecake or cream cheese component, right? So then you can like leave that part out, okay? And you could do that different every time, right? So one recipe can make multiple different things that are like relatively similar, but definitely serve different purposes, okay? Cool. All right. Thanks for joining. Keep learning.